Okay. Well, hello, and thank you for joining us for the last of 4 webinars that are part of the Core 7B Gulf of Mexico Research Initiative, Synthesis and Legacy Efforts. This session has 2 moderators. I am Cecilia Mauritzen, an oceanographer at the Norwegian Meteorological Institute, and I'm also a member of the GoMRI Research Board. And we have Eric Chassine, who is director of the Center for Ocean Atmospheric Prediction Studies and professor of oceanography at Florida State University. Now, questions can be entered anytime during the webinar in the chat box located at the bottom or right of your screen. Eric will consolidate a subset of those questions for the speakers to answer at the end of the 3 presentations. The 7B synthesis effort from the Gulf of Mexico Research Board is called Modeling for Synthesis. We wished to find a way to answer the overarching questions, the questions that motivated the Gulf of Mexico Research Initiative in the first place, such as what are the impacts of the oil, of the dispersed oil, and the dispersants on the ecosystems of the Gulf of Mexico? How do such events impact human well-being and the functioning of communities? What can be done to mitigate the gravest impacts? How can we be better prepared next time? These questions cover a multitude of timescales. What is done in the first few hours can impact societies for years to come. And vice versa, how well communities are prepared can affect what happens in the first few hours. And these questions cover a multitude of scientific disciplines that use a variety of methods. But to answer the overarching questions, we need to speak the same language. And the Modeling for Synthesis effort is an attempt to do just that. We have chunked the problem down to 4 subdomains. It's the health, it's the ecosystems, it's the socioeconomics, and the physics. This week's mission is to identify the key feedbacks and linkages within the physics box and between that box and the other boxes. The 7B synthesis process will continue through the summer and fall as we bring the subdomains together and get ready to have dialogues about the overarching questions and all the what-ifs. Now, for today's session, Sea Grant has collected a set of questions about physics that are of interest to the general public. So Monica Wilson of Sea Grant will briefly explain the origin of these questions.
Thank you, Cecile. So as she said, my name is Monica Wilson. I am the Florida Sea Grant representative of the Sea Grant Oil Spill Outreach Team. The team aims to increase the use of oil spill science by people whose livelihoods depend on a healthy Gulf of Mexico. Our team continuously engages with people in small and large group settings to learn about their oil spill science related questions and concerns. And the questions that are shared here represent our target audience input between 2014 and 2018. Our team solicits input multiple ways: one-on-one or small group meetings with target audience members, input sessions with large groups of target audience members, and through evaluations completed before and after oil spill science seminars and workshops. The questions that were pulled, these questions that you see here were pulled specifically for this core area synthesis, and they don't represent the entirety of the data Sea Grant collected for the program. Thank you.
So these questions, yeah, you can see them on the screen, but they include like where did the oil go? Where are the biggest deposits today? How long did the oil take to reach those deposits? What beaches are affected? How much is buried on the seafloor? And here's a good one. Could a big storm bring the oil up to the seafloor, up into the water column, and start the process all over again? Did any oil make it into the organisms living in the water column or on the seafloor? What happens to the oil over time when dispersants are applied? What are the natural organisms that decompose hydrocarbons, and how can we increase that process? And was it possible to track the oil with numerical models? If not, can we do it better now? Yeah, so we have a lot of interesting questions, and we have a good set of speakers lined up here today. The meeting today will be followed by a virtual seminar or a virtual working session on Thursday from 10 to 12 Eastern Time. And during that working session, we have invited guests that will be discussing the beginnings of the conceptual model framework. So if you're interested in listening to that virtual session, please type your email address into the chat box, which still should be found on the bottom right-hand side of your screen. So, on to the presentations. Our three presenters are Dr. Deborah French-McCay of RPS ASA, which used to be called Applied Science Associates. We also have Professor Bill Dewar of Florida State University, and we have Professor Michael Stuckel, also of Florida State University. So first, I would like to invite Dr. Deborah French-McCay to introduce herself and describe her vision on modeling oil transport, fate, and exposure for evaluating the evaluation of environmental impacts.
Good morning or afternoon. I'm Deborah French-McCay. I've been at our ASA and now RPS for 35 years. Today, let me just get this into presentation mode. Can everyone see this?
Debbie, we see presenter slides, not full screen.
Really? Sorry. Last time it worked just fine.
But it is probably okay.
All right, to save time, let's not worry about it, yeah.
Yeah.
Okay. I'm going to give you an overview of a model system we've been developing over the past 3 decades at ASA, and now as part of RPS, which we call CIMAP. The system includes a series of models. First, a physical fate model that calculates the oil trajectory and concentrations of oil and their constituents in space and time. And then an exposure model that records the concentrations and environmental conditions that are experienced by organisms in various habitats over time. An oil toxicity model that evaluates biological effects. And in the case of a blowout, we have a near-field model that evaluates the trapping of the buoyant plume and calculates the droplet size distribution of the oil released to the far field oil fate and transport model. So there's a series of outputs that are depicted in orange here, and depending on the problem, we'll use different parts of the system on different types of outputs to evaluate the issue. As I mentioned, we've been developing this over many decades now, so I won't go through all of this, but I'm going to focus on the Deepwater Horizon, our most recent work. We worked for NOAA as part of the natural resource damage assessment, developing and applying the model to Deepwater Horizon. And then we've been continuing our work, some of it for BOEM and other ongoing projects to evaluate the fate and effects of the spill. So for the Deepwater Horizon, we used our oil map deep model. Can you see my mouse? And this calculates the buoyant the buoyant phase of the plume and the trapping height. And so we release droplets into the far field model from the trap height in a series of droplet sizes. And then the far field model calculates the rise of the droplets and the transport of them and all of the other processes that are going on in the physical fate model, and then it moves into the other models. So our models are Lagrangian. So we break up the oil into the different parcels called Lagrangian elements and track them as they move through the system. So we can track the weathering, we track whether they're droplets or adsorbed to particulates or floating. And then we also track the dissolve phase. We similarly use Lagrangian elements to track the organisms so that we can keep track of their exposure history over time. And then we read in the environmental data from various gridding systems. We also keep track of different components in the oil. We divide them up by volatility first. And so they're in a series of boiling cuts, essentially, which characterizes their vapor pressure and tendency for evaporation. And then we also group them by solubility using the octanol water partition coefficient, log KOW. So we're separately tracking alkanes and other aliphatics from the monoaromatics and the PAHs in various groups. The most important processes in a deep water blowout, it really starts with the droplet size distribution that is released from the near field model. If the droplets are greater than about 500 microns, they're going to surface very quickly in about a day, in the case of deep water. And however, if the droplets are less than 100 microns, they're likely to stay down at depth and not ever surface. And then there's an array of sizes in between that are going to rise at ever slower rates and move farther away from the spill location. So we focus on the droplet sizes and then all of these various processes. Once the oil reaches the surface, again, it's the turbulence and the droplet size distribution that drives the results. The more turbulence, the smaller the droplets, and then the more likely they are to stay down and disperse. And then there's, of course, more exposure to water column organisms when this oil is dispersed. And dispersant enables this process. So we ran the Deepwater Horizon spill with a number of assumptions and inputs, but we also tracked the change in the release volume to the environment over time. As you remember, on June 3rd, they added a cap, and then they were able to recover a large fraction of the oil. There were also releases from both the end of the riser and small holes in the kink that started in May. And so these small holes created small droplets, whereas out of the end of the riser, without dispersant, there were large droplets. And then, as you note, the dispersant volume applied daily varied tremendously over time. The blue is the surface dispersant, the purple is the subsea dispersant. So we calculated on a daily basis the dispersant to oil ratio, and therefore the droplet size distribution, which showed considerable variability. There are some various assumptions I don't have time to get into, but you can assume that the dispersant was completely mixed in the plume, and then get a droplet size distribution that varies depending on the amount of dispersant. This is time here on this axis. This is the size distribution on this axis, and the volume fraction of the mass is on this vertical axis. So we start with very large droplets before the dispersant is applied, moving to smaller and smaller droplets later in the spill. So this changes the entire fate of the oil. However, what we determined was, in order to account for the surfacing oil that occurred throughout the event, even into July, and looking at the release from the plume, we accounted for a partial treatment of the oil with the dispersant. And in that case, there's two different size distributions, one for the dispersant treated oil, that's small droplets that would have formed the deep plume, and then large droplets that surfaced rapidly and formed the surface oiling. We also spent a considerable amount of effort looking at transport from currents. There were an array of ADCPs out on the slope, and we used that data. And we also used as input a number of different hydrodynamic models. Each of these hydrodynamic models moved in the oil in varying directions. What we found was that using the ADCPs as an indication of what happened, when we interpolated the ADCPs, we get a trajectory below 40 meters that goes out to the southwest as observed. And we found that the HICOM models did a much better job than the other models. And the best analysis came from using the FSU HICOM model, which did well above 40 meters and quite well below, although there were slight details or somewhat detailed differences in the trajectory, but generally agreement with the observations. This is a map of the remote sensing from SAR, from synthetic aperture radar. It's the cumulative days of SAR oiling based on the SAR observations. Our model predictions show the same focus of oil distribution in this area off Louisiana, Alabama, and so on, but also showed scattered sheens farther away that were not observed from the remote sensing. In comparing the model predicted amount of oil in the water, so this is volume of water, oil floating on the water. The black line is the amount of oil. The orange line is the amount of mousse, including the water. And then these various symbols are the different remote sensing data that are based on observations of color or other indicators of thickness. And so you can see there's a lot of variability based on the remote sensing, but that there's general agreement between the model and the remote sensing estimates, the amount of oil floating on the water. We also compared the model predictions of shoreline oiling. The orange are the match to the observations based on SCAT. And the red are areas where the model predicted that oil should have come ashore, but they weren't actually observed there. And the yellow are areas where there was oil observed onshore, but the model did not predict it. And this is using the FSU HICOM. This map shows you the distribution of oil on the shoreline and also in the sediments, both in the nearshore area and the deep water area, again, using the FSU HICOM. So, summarizing the water column concentrations is a little difficult because it's so dynamic, as you can see from these snapshots. So what we did was, since most of the observations were within 25 kilometers of the wellhead, we made a box around the wellhead and compared the measurements to the model predicted concentrations and looked at the probability distributions of each and found that there was good agreement in the magnitudes of the concentrations, even though that the trajectory wasn't exactly aligned with the samples in space and in time. So in conclusion, we obtained reasonable agreement for the floating oil and the shoreline oiling. The water concentrations that were predicted by the modeling were of the same magnitude as the observations. The subsea dispersant was effective on the treated oil, but not all of the oil was treated with dispersant, which accounts for why there was so much oil floating around. And then the model predicted that the highest concentrations were near the trap height, as observed, and that most of the BTEX and soluble alkanes dissolved at depths near the trap height, and any that came up with the oil evaporated rapidly right at the wellhead area. And the PAHs only partially dissolved as the oil rose, and so the exposures occurred both in deep waters and at the surface. I'm just going to briefly touch on the exposure model and toxicity model. So we use Lagrangian elements to track the exposure. So we classify organisms based on their behavior, whether they swim, drift with the currents, or remain stationary. And then we define their vertical zone and swimming speed and other kinds of behavior. And then the Lagrangian element tracks the concentrations over time, as well as temperature and light exposure. And from that information, we calculate an exposure duration, mean concentration exposed, and we compare that to LC50s or other endpoints to calculate the effects. One of the important considerations is that the LC50 is dramatically higher at a higher concentration or less toxic for very short exposures. So our model is accounting for duration of exposure for that reason. And there's also probably a temperature effect to this. We've validated the oil toxicity model. First, I compared them to oil bioassays, and that's written up in this paper, and for 24 different sets of data, and got good agreement. And then we also applied it to the North Cape oil spill, which occurred in January of 1996, where lobsters were dramatically impacted. The field estimates were based on quadrat sampling, and it was that 9 million lobsters were killed, and then the model estimate compared well with 8.3 million using a species-specific LC50. So in conclusion, the model has been developed over many years. It's been peer-reviewed a number of times, and then we have publications and been validated. It's really important to consider the dynamics of the system, not only in the oil fate, but the exposure and toxicity in order to evaluate the impact. And we've applied this model system to many different kinds of problems such as hindcast of different spills, natural resource damage assessments. We've done many risk assessments, both for spill response planning and as part of NEBAs, or now called SEMA. And most recently, we did a comparative risk assessment of various response alternatives, including dispersant use at a wellhead for blowouts, to evaluate the net benefit of using dispersant. And that's it.
Thank you so much, Deborah. This was incredibly instructive. And Professor Dewar, I would like you to take the floor now, so to speak, and talk about oil from the first to the last kilometer and everything that happens in between. Your turn. Are you unmuted?
Hello?
There you are.
Can you hear me?
Yes, vaguely.
Very good. I shall try to do justice to this title. So my hope today is to talk a little bit about what GoMRI has added to the problem of what happens to a deep oil blowout. I think quite a bit of progress has actually been made under the GoMRI umbrella. My particular experience was with the Carthy Consortium out of the University of Miami, listed here. But I would like to acknowledge the broader effort of the Gulf of Mexico Research Institute, which I think probably in the neighborhood of hundreds of physicists have been supported in this initiative and have all added significantly to this problem. All right, so schematically speaking, here is what we have. We start down here where a deep blowout would first occur around a wellhead, generating a buoyant plume of oil, which rises through the water column. In the process, numbers of things happen. The plume itself is dynamically active. It interacts with the background flow fields to displace a significant part of the plume effluent laterally on the water surface. Once it gets to the surface, then a number of processes happening can happen to it: weathering, biodegradation amongst them, evaporation. There's interactions with the weather, the displacements due to winds and surface currents, as well as waves, are responsible for moving the oil around. And then when it gets to the very near shore, it starts interacting with a unique environment, which is found in the very shallow coastal environment. So those are the, in a few words, the chunks that we'll be talking a little bit about today: the near field, the not so near field, the in-between, and then the last kilometer. We start then near the wellhead, the first kilometer. And I think GoMRI has made a couple of surprising contributions to this problem. This is a classic problem: buoyant plumes and fluid from a sustained source. But what GoMRI added to the problem, I think, were two main new results. The first one, rather surprisingly, is the fact that the Earth is rotating is a significant input to the plume behavior itself. That's what's shown in these plots over here. So in this column, we have numerical simulations of what a plume will do, being released here in the middle of the grid. This blue over here is essentially what the oil would be doing in the water column. Over here on this side are plots of the buoyant anomaly associated with the plume. There are two columns. As we move down the column in this direction, the importance of rotation increases. We estimate that the Deepwater Horizon event was somewhere around in here, about halfway down the diagram in terms of the parameter which describes rotation. What you can see for the non-rotating case, which is a classic case, is a rather standard, nice vertical plume with a separation peel-off layer at a location that can be written down in terms of the parameters of the problem. What we found is that as you start to increase the effects of rotation, the plume begins to become suppressed in terms of its ability to penetrate in the water column. In this particular case, which is not an unreasonable measure of rotation for the Deepwater Horizon, you see the effects on the plume are rather dramatic, and it resulted in what we referred to as a precession of the plume. The plume becomes no longer a simple vertical entity, but instead rotates around the wellhead in time. And the distribution of oil that results is, as you can see, changed quite a lot. So this ends up being of significance with regards to the spreading layer, which is where a lot of the subsurface oil that exited the Deepwater Horizon event was trapped. These are results in the absence of a background flow. If we include background flow, we see things which are reminiscent of that do penetrate through the inclusion of background flow. This is the data that was obtained during the Deepwater Horizon event. These are various measures of oil concentration at depth emanating from the wellhead. They follow what we heard about a little bit ago is the southwestern trajectory of the oil. And you'll notice that along that pathway, there's a considerable amount of variability. If we turn on a deep oil spill and allow for a crossflow, here is a numerical simulation. This is a flow which is moving from the left-hand side of the page to the right-hand side of the page, unfortunately in the direction opposite to the southwestward trajectory that we see in the data. But nonetheless, you'll notice that the dispersion is pushed along by the flow, but does take on its own character. So for example, we have down here a collection of what would be oiled water, which under its own dynamics is separated from the main plume and is now marching off, in this instance, to the southeast. So this is evidence of the plume itself being dynamically active. That has to do with the extreme buoyant anomaly that is oil. And again, it's something which I think wasn't expected and GoMRI has shown. If we go to the in-between, now this is the surface, when oil gets to the surface in the vicinity of the plume wellhead. This is prior to reaching the coast, still in the deep water. The thing that GoMRI has demonstrated quite conclusively is that fronts are critical to the subsequent surface distribution of the oil. This is looking down at the Gulf of Mexico, and what one notices is that the oil has collected along a density front on the surface. So the surface dynamics are very effective at taking the oil, which itself is a buoyant material, wants to float on the surface. And because of the dynamics which are characteristic of the upper 100 meters or so of the ocean, it is very effective at concentrating it into these thin stripes. This is looking down on the Gulf of Mexico on a smaller scale than this plot over here. And we see another example of the oil accumulating into streaks, but this time what's happened is that the smaller surface gravity waves have organized the very near surface flow into Langmuir cells. These are the well-known phenomenon of windrows, which people who've gone to the ocean and to freshwater lakes for that matter are very familiar with. And we see that what's happened is that the oil has been accumulated by the Langmuir circulations into these very intense streaks. So the oil is naturally concentrated in given locations. The appreciation of this and the dynamics of the surface layer, which are responsible for it, have been quite considerably illustrated by a number of efforts during the GoMRI, by the GoMRI scientists. And this is of some considerable practical significance. A demonstration of that is what I'm going to show next. This is due to some of our friends at the University of Delaware, Denny Kerwin, principal among them, as well as Greg Jacobs at NRL. This is an example of how informing collecting ships with this information can be quite beneficial. So quickly, the red ship and orange ships are two ships that are supposed to go and collect oil. They're equally capable. The red ship is provided with information about where the oil is. The orange ship is given that information as well, along with further information about how the surface convergence is taking place. So it's trying to exploit this information about surface fronts. And so we have, as time goes on in this example, here are the ship tracks. How much oil can each of these ships collect? And this is running a couple of times. Down here, the total amount of oil that's being collected is plotted up as a function of time. Don't pay any attention to the dashed lines in this. That's a different oil collection scenario. The important lines for our discussion are the solid lines. The blue line is the ship that knows about the convergence rates, and the red line at the bottom is the one that does not. And you can see that knowing about surface convergence is a great benefit to oil collection. And in fact, in this experiment, 8 times as much oil is collected by that boat as opposed to the one that doesn't know about surface convergence. So practical significance of the surface phenomena is quite clear. Now, if we go to the final kilometer, which is the very near shore, that's something that I'm showing schematically here. This is not an oil distribution. This is from a numerical model, a very high resolution numerical model in the very near shore area. Here is the Mississippi River outflow. It's showing up here as blue, and that's because this is, this is vorticity. It would also correlate quite a bit with the freshwater anomaly that's associated with the nearshore environment, particularly around a very strong effluent river like the Mississippi River. As a result of that presence of freshwater intruding into the otherwise relatively salty Gulf of Mexico, very strong fronts again are formed, but this time rather than from open ocean set of dynamics, from the near coastal dynamics associated with the freshwater environment. I know that this is something that's been worked on by our very good friends at UCLA, Professor Williams and Dr. Molemaker, and also University of Miami. Dodi Karafalou has been very active in this area. These fronts are quite good at shielding the coast from direct penetration by tainted water onto the coast. Another thing which is also implicit in this is the wave zone, the wave dynamical coastal zone, which is also itself a very strong influence on the transfer of water from the very, very near shore onto the coastline, involving other well-known phenomena like rip currents, for example. So anyway, those are the 3 areas, first kilometer, last kilometer, and what's in between. I think that's safe to say as a result of GoMRI funding, a lot has been learned about the distribution of an oil-like substance as it moves from the wellhead onto the coast, and some of the things were simply not known before. These are important for a number of practical reasons, as I've tried to point out here. And one would hope the synthesis activities that something like Core 7B is pointed at would work to exploit this information. Some of the examples that I've seen of ongoing synthesis activities, it looks like everybody is working very hard to couple everything to everything else, and oftentimes using very simple models to do that. This is for very good reasons, because in this effort to synthesize widely disparate areas, we are making our first attempt, and so starting with simple models makes a lot of sense. But I would hope in the synthetic activity that it would not be forgotten. A lot of advanced knowledge has now been learned about oil spills as a result of GoMRI efforts, and I hope we can work to include that knowledge in some of the later generations of synthesis models. So with that, let me turn the floor back to our distinguished moderators.
Thank you very much, Bill, and your challenge is well received. And now it's Professor Mike Stucco also of Florida State University, and his title is Integrating the Roles of Microbes, Currents, and Sedimentary Processes in Transport and Transformation of Oil Spill Hydrocarbons. Welcome, Mike.
Thanks. All right, can you see my screen?
Yep.
All right. I'm Mike Stucco at Florida State University, and I work with the GoMRI-funded consortium to simulate oil microbial interactions in the ocean. I'm going to try to build onto the hydrodynamics that Bill just expertly presented to us by explaining how marine microbes can alter hydrocarbon transport and transformation. So Bill just explained to us the ways in which currents affect the distributions of oil in the water and where it winds up. So I'm not going to go into this in too much detail, but basically we've got oil that gets released and it rises depending on the buoyancy of the oil. Some of it breaks into droplets. It spreads into a deep water plume, and some of it rises to the surface for a surface slick, and where it's going is largely determined by the currents. But the transport and fate are also determined by the buoyancy of the oil itself, by the interaction with sediments, by photodegradation and evaporation, by incorporation into marine oil snow aggregates, and by microbial degradation. So I want to just briefly talk about the ways in which each of these can affect oil transport. So first off, buoyancy is dependent on the actual composition and size of the oil droplets, what oil makes them up, and that in turn is largely affected by the role of microbes that live associated with these rising oil droplets and break some compounds down into others. And then, of course, the oil interacts with sediments on the near shore depending on wave energy and the sedimentary composition. And when the oil makes it to the surface, it gets photodegraded and evaporated, which changes the composition of the oil and makes some forms of the hydrocarbons more bioavailable to microbes. One of the interesting findings of a lot of the GoMRI funded research was actually that a lot of the oil got incorporated into what we're calling marine oil snow aggregates. These are some figures from Uda Passau's work. And basically these are aggregates that vary in size from about 1 millimeter to greater than 1 centimeter in size. They're produced by exudates from marine microbes. And these marine snow particles were collecting a lot of the oil and then sinking into the deep sea, thus transporting the oil from the surface ocean down onto the benthos. And of course, microbes are degrading a lot of these hydrocarbons. And it's important to keep in mind that microbes are not necessarily just chewing entire hydrocarbon molecules, but rather what they're doing is, for the most part, releasing enzymes that specifically cleave different parts of these hydrocarbons. And when they break them down into smaller molecules, they can then take up and degrade some of those molecules. But this is really a species-specific interaction because different organisms produce different compounds that act against some hydrocarbons more than others. So I want to talk a little bit now about the CSOMIO approach that we're taking. We're using multiple different components and integrating them into one larger model to understand oil transport and fate following Deepwater Horizon oil spill. We've got a physical model that determines the currents and where everything goes. Embedded within that, we've got an oil transport and weathering model, as well as a sediment transport model and a microbial genomics model called GENOME that I'm going to talk about in a bit more detail in a couple minutes. But first off, our physical framework is the Coupled Ocean Atmosphere Wave Sediment Transport Model, or COAWST-ROMS modeling system. It models atmospheric interactions on the ocean, and particularly includes wave interactions. The waves then interact with the composition of sediments, the sediment grain size at the bottom of the ocean. And so we generate realistic surface waves and sedimentary interactions. And so the sediments themselves can then interact with the oils and form oil aggregates. And then we've got, coupled into this, we've got an oil transport model similar to the one that Debbie talked about before that treats some compounds of oil as dissolved in the seawater and others as Lagrangian superparticles that are advected by the water, but also then colonized by different microbes. And then interacting with this, we've got a novel model called the GENOME model. The GENOME model was really developed to address some of the limitations of typical functional group models. So functional group modeling has been used for a very long time to understand marine ecology and marine biogeochemistry. The way functional group modeling typically works is you have a few basic functional groups of phytoplankton. So you may have one group that represents diatoms, another that represents nitrogen-fixing organisms, a third group that includes mixotrophs, and a fourth that includes calcifiers like coccolithophorids. And all of these interact and mediate some of the basic biogeochemical transformations in the ocean. But one of the big issues with this is that these are not individual species in the ocean. They're hundreds of different diatoms, hundreds of different nitrogen fixers, thousands of mixotrophs, and a ton of different calcifiers. And all these different organisms are evolving as a community in the ocean. And so a typical functional group model cannot adapt to a changing functional group community, and it often lacks organisms for some processes. For instance, the vast majority of functional group models are not going to have any organisms that degrade hydrocarbons. Furthermore, these functional group models have, at best, if anything, indirect links to genomics and transcriptomics data that we're now rapidly generating in the field. So to address these limitations, we developed a fundamentally new type of model called the genome model. The genome model includes hundreds of different organisms, and first we assign each of these organisms a random size. The size can range from anywhere to less than a micron to anywhere from 100 to even 1,000 micron in size. And the random size that an organism gets assigned affects some of its properties. The next thing we do is we assign to each organism a number of genes and then randomly assign the genes themselves to these organisms. So these genes can include things like genes coding for breakdown of long-chain alkanes or methanotrophy, but they also code for some of the basic biogeochemical capabilities of the plankton community, including photosynthesis or nitrate uptake. So once we've assigned all of these genes and sizes to organisms, those sizes and genes that they've gotten affect how they interact with the environment, including what substrates they can utilize, what biogeochemical reactions they can mediate, and also what their growth rate is going to be as a function of size, what their nutrient kinetics will be, what their sinking rates will be. And so once we've defined all of this for all these organisms, we can put them into a general circulation model like HICOM or like COAWST-ROMS. So we insert these organisms into this general circulation model, and they get advected around by the physics. And as they're being advected around, the differential equations that govern their growth and substrate utilization determine how much they grow, what substrates they're utilizing, and how much, for instance, nitrate they're taking up, or how many hydrocarbons they're breaking down. So as they're being advected throughout the ocean, they're mediating all of the biogeochemical transformations that we find in the real ocean, and they're breaking down different types of hydrocarbons. This genome model has a lot of benefits for studying oil microbial interactions, because it simulates marine biogeochemistry and hydrocarbon degradation as an emergent property of microbial community dynamics. So we're not specifying a priori how we think these organisms should interact with oil. The interactions between the oil and the way that they break down hydrocarbons is an emergent property of the model, which means that we can take this and simulate different oil spills in different places around the world and expect the model to figure out for itself how these organisms and communities should adapt over time. It also allows community evolution in response to environmental perturbations, such as the oil spill. So the way this works in the Gulf of Mexico is we've got background communities that are existing on hydrocarbons that are produced during natural methane seeps. And once we get an oil spill blowout, some of these communities rapidly evolve to the new conditions. The model also allows us to directly predict metagenomics and metatranscriptomics patterns that can be then compared to in situ measurements, which gives us a powerful ability to actually validate our model against in situ measurements. And finally, the genome model is embedded in hydrodynamic models, allowing fully coupled investigation of transport and fate of hydrocarbons within the same sorts of models that Debbie and Bill have talked about previously. So this shows a basic 1D simulation of genome after the Deepwater Horizon oil spill blowout. So what you see here is a graph in time. On the y-axis, we've got depth. On the x-axis, you have time. The different plots at the top are showing different substrates. So first, we've got a typical saturated hydrocarbon. You can see the first maybe quarter of this graph is prior to the blowout. Then after the blowout, we suddenly see an increase in hydrocarbons at the deep water plume location and at the surface location. The plot to the right shows aromatic hydrocarbons, which are more difficult for the microbes to break down. And then you see ammonium and nitrate continuing on the top. And at the bottom, we've got some of the different microbes that exist in the model. So if you look at microbe 26 at the bottom left here, you can see this is a microbe that's actually very efficient at breaking down the saturated hydrocarbons, at breaking down things like simple alkanes. So what we see is it grew up rapidly after the oil spill blowout, and then it was able to actually rapidly draw down the saturated hydrocarbons. But the aromatic hydrocarbons lasted longer in the deep water for the simple reason that they're much more difficult for the microbes to break down. We see this microbe 33 is a microbe that seems to do particularly well in the surface ocean, possibly because it needs warmer temperatures, and was actually able to break down some of the aromatic hydrocarbons that had started to build up in the surface ocean. We also see, if we look at ammonium, that the ammonium concentrations actually built up in the water column while nitrate was being drawn down because these microbes needed to take up nitrogen when they were getting their carbon from the hydrocarbons. So this is our basic 1D simulation, and we're currently in the process of putting it into the COAWST-ROMS framework so we can do full 3D simulations of the interactions between oil and the microbes. And of course, this work was done by myself and a lot of my colleagues at multiple institutions working with the CSOMIO project. Particularly, a lot of this was done by Victoria Coles and Jiazi Wang at Horn Point Laboratory. And that's it for me.
Thank you so much, Mike. I'm amazed by these three extremely good presentations and also keeping time so well. So I think we actually have time for a little dialogue here. So I leave the word to Eric. And then we'll figure out how to do questions and answers online. Eric?
Right, so if you have any questions, please put them in the chat box. So right now I have a couple of questions that were raised, one for Bill and one for Mike. So I'll start with Bill from John Shepard. Assuming that we can't model the small-scale convergence in real time, what observational data would be needed to enable the superior collection strategy? Bill, did you hear?
Hello?
Yep, we can hear you.
Okay. As one has come to expect, the perceptive and distinguished Professor Shepard has asked a very good question. I think, well, with regards to the simulation that was shown there, that was a simulation, the surface convergences of which were not not associated with Langmuir cells or any of the very, very small scale fronts that we see. Those were actually more the fronts associated with the preferred locations for submesoscale that are set up by, in this case, the mesoscale. So a lot of the locations where we expect fronts to be set up are kind of predetermined by the mesoscale field. The way the information and the strategies that are employed by the ships are set up is something that I would refer you to Professor Kerwin for further information about. The long and the short of this answer is, I think with a well-constrained model that's going to work to get mesoscale and mesoscale structures in roughly the right locations will go a long way to providing information about the convergence rates that will end up being useful to surface oil collections. So maybe this can actually, this might actually be something that can be done in real time.
John, does this answer your question?
Hi, yeah, up to a point. I think the interesting question then becomes, you know, where is the scale separation where we could expect to synthesize model and data to provide useful information, and you know what scale is going to be too small, so that even if it happens, we can't produce the information for the ships to work on.
Fair enough. Good point. I don't know that I have a good question or good answer to that question at the moment.
Any follow-up comment or on this question? If not, CJ had a question for Mike. Mike, so oils vary around the world. So how do we connect with all the different type of crude oils?
So when we've been developing the genome model, a large part of our goal has been to keep it as generalizable as possible. So rather than specifically tuning it to the type of oil that's being released during the Deepwater Horizon oil spill, we're basically developing it such that microbes generally utilize something like 8 to 10 different classes of hydrocarbons. So some of them are very simple things like methane, ethane, propane, but then we've got longer chain alkanes, we've got polyaromatic hydrocarbons, and a couple other classes. And so if we know what types of hydrocarbons are released during different oil spills, the only thing that we would need to change when running the model in a different area following a different oil spill would be the actual concentrations of those different compounds that are being released. So we would need to figure out for whatever particular oil spill occurred, what proportion of that oil was methane, was ethane, was propane, was short and long chain alkanes, or polyaromatic hydrocarbons. And then once we know that, we can run the model and the microbes will degrade that oil depending on essentially the bioavailability of those different compounds. Does that answer your question?
CJ? Yeah. Okay, well, in the meantime, John has an interesting question. Is it possible that your bugs may adapt to imperfections in the model representation rather than to the real world? Could that be a problem?
Of course. I mean, anytime we're developing biogeochemical models like this, it's there's always the possibility of sort of overfitting to what's going on in the model. In this type of a our model, I didn't really go into the full details, but it's really a stochastic model because we're randomly generating bugs. And because we're randomly generating all of these different microbes, there's no guarantee that we're finding a single solution. So really, when we run these models, we do a whole bunch of different simulations. So we'll do an ensemble of, say, 10 different simulations with the exact same physics, exact same oil release, but with different randomly generated bugs at the beginning of the model. And so our hope is that because we're using a bunch of different random communities that then evolve to the conditions that are as realistic as the physical oceanographers can give us, that the similarities between runs will be based on the model actually capturing what's really going on in the ocean, while differences between the runs may be a problem of getting, for instance, super microbes that maybe don't really exist in the environment. So our hope is that by running these multiple different simulations and looking at the similarities between them, we will have less of an issue than typical functional group models, which would exactly tune and say, we think that this particular microbe, say Alkanovorax, is chowing down on alkanes at this rate. We're not prescribing that, and so we hope that that gives our model more generalizability. But it's always a possibility that your model is getting the right answer for the wrong reasons. And that's something it's never possible to rule out. The model is a hypothesis, and you can never prove that it's true. You can test and see if it's wrong, and that's really all you can do.
Thank you, Mike. So Debbie put forward a question. How can advances in remote sensing be used to inform response in concert with trajectory modeling? For example, during the Deepwater Horizon, the modeling was used to direct overflight, and then the responders were directed to the fronts where the oil was collected. So I think we're talking about real-time remote sensing. Debbie, do you want to elaborate a little bit on?
Yeah, so, I agree that the fronts are really important and, other small scale features, but we're going to have a hard time predicting the exact locations of those. And in recognition of that, the typical response is that trajectory modeling is used to direct generally where the oil would be, and then overflights are used to determine exactly what it looks like and then to direct the boats. So we've gotten much better at that. Originally it was fly in a helicopter and make sketches, and then during Deepwater Horizon there were overflights with remote sensing imagery that was used. So that's, with the overflight, you're close enough as opposed to in a satellite image to actually resolve these smaller features. And so a lot of work has gone on in remote sensing over the years, and it seems like that could be used in concert with the trajectory modeling to direct the responders. And I thought maybe Bill wanted to comment on that. I'm just throwing that out as an important part of that.
Bill?
Yeah, can you hear me?
Yes.
Well, I mean, what can I say to that other than I would completely agree? Data source that can be used in order to help constrain where the fronts would be. At scales at which at some point it's not going to matter too much if you get to be a finer scale, because when you put the ship there, direct the ship to some particular location, then people on board are going to take over control. So getting a ship close, that's where the payoff can be the greatest, and I think that's where this information is likely to be the most useful.
Could I add something? This is Willie.
Go ahead.
Yes, during our GoMRI project, we actually did an interesting field study using the Taylor energy platform, the oil around energy, Taylor energy. And so we used, the newest, very high resolution satellite. So we waited with that. We coordinated with the times that we expected to have that data. And we had a small boat standing by, larger boat, the Walton Smith from University of Miami standing by, and also drones. The drones seem to be very effective in giving more detail to direct the boat.
Okay. For Mike, what do we know about, I'm sorry, the interaction of dispersant with microbial effectiveness?
Okay, we know that the dispersants definitely have an effect. The effect it seems to vary by different groups. I mean, there's been shown to have some studies have shown there to be an effect on zooplankton. Some has shown there to not be an effect. Some studies have shown there to be an effect on protists and bacteria. Some studies have suggested that there's not. I would say that the best consensus right now is that the dispersants are a deterrent to some of the oil degrading microbes that comes out of, I think it was Samantha Joy's group, had evidence that the microbes were breaking down the oil less effectively in the presence of dispersant than without dispersant. But I think there's still a lot that's unknown about the effect of dispersant. There's a lot of variability study to study in these different projects that have looked at specifically the physiological response of different microbes to oil and dispersant. And part of that's driven by the fact that most of the organisms in the ocean that are actually degrading the oil are not organisms that we have in culture. There are a few very prominent taxa that we have been able to culture, and so those are the ones that have been generally used for manipulative studies. But in the field, there's actually a really diverse community, so we don't have a great handle on it. I would say my best guess is that the dispersants are actually bad for the microbes, but I think there's a lot more research that's needed on that topic. Thank
you, Mike. Tom, you had also a dispersant question. I mean, did this answer or address some of your question? Okay. Thank you. So Tom said yes. So I think, we're getting, past the hour now. I think we need to wrap up. Elena or Cecilia, do you want to say a few words to closing? I mean,
well, I just want to thank everybody for a great webinar today, and again remind those who want to join or listen to the discussions on Thursday. That'd be really great, and those who want to follow the process going on, let us know. We don't quite know where this ends, but we have great faith that if we talk together from all these different disciplines, we will all learn something. So thank you again, and be good. Bye-bye.